Our next speaker will be Julia van Etten, if I correctly pronounce your name. And Julia is from the State University of New Jersey, and she will present her talk about genetic transfer in action, modeling DNA acquisition within extremophilic microbial community. A space is yours, Julia.
Thank you. Let's get this.
Yeah, we see your screen. Everything is okay.
Okay. I can't see myself, but as long as you can see me. Okay. So thank you. I'm Julia. I'm a fifth year PhD candidate in the Bhattacharya lab at Rutgers University in New Jersey in the US. And today I'm going to be talking about modeling genetic transfer through an extremophilic microbial community. And so I'm going to discuss the Cyanidiophyceae red algae, their high instance of gene sharing, and some new computational methods we're using for modeling it. And I'm glad there was another presentation where the speaker discussed Cyanidiophyceaen, because I won't go into all the details here, but basically, these Cyanidiophyceaen algae are the smallest photosynthetic eukaryotes, and they've become adapted to environments previously dominated by prokaryotes. So they show us that organisms of eukaryote level complexity can involve polyextremophily by many of the same mechanisms that prokaryotes use. And not only do they survive, but they thrive in some of these habitats. And so if they can dominate some of the harshest environments on Earth, what does that mean for the limits of eukaryotic life and the search for complex cells in similar locations elsewhere in the universe? And so when it comes to biomass, the Cyanidiophyceae are dominant organisms in the thermoacidophilic locations like the hot springs of Yellowstone National Park, which is where a lot of the research for our projects take place. And here, these red algae make up over 80% and sometimes over 90% of the biomass. And they're found in these thick green biofilms within creeks and streams, acidic subsurface soils bordering the water, and some groups even form endolithic populations within the rock crevices. So they've really dominated these extreme environments. And so if we zoom into this creek biofilm, we would see eukaryotes, mainly the Cyanidiophyceae. Here is an example, the genus Galdieria. We would also see many bacteria and archaea living in close association with them. Some of these prokaryotes are releasing exogenous DNA into the environment. And we would also see many viruses among the cells of the biofilm. And so these dynamics create a lot of opportunity for gene sharing to occur between closely related organisms and even across various domains of life. And just some more background. So the Cyanidiophyceae have had over 1 billion years to adapt to the extremes of temperature, pH, salt, high toxic heavy metal concentrations like arsenic and mercury, and fluctuating light availability. And so we see evidence of this adaptive history throughout their genomes, which are gene-rich, intron-poor, and highly reduced. And over time, genome reduction can often lead to the loss of important genes and the accumulation of deleterious mutations, especially in asexual organisms. But I will add that a paper just came out that showed sexual reproduction in these algae. So anyway, so one way to counteract negative changes that come along with genome reduction is to compensate for gene loss with a process like novel gene origination. And the quickest way to originate a new gene is to simply receive it from another organism that has already evolved it and optimized that gene to encode a beneficial function. And so many studies in recent years, including a few that I put on this slide, they've shown that the Cyanidiophyceae, which include Galdieria and Cyanidiophyceaen, have received an assortment of horizontal gene transfers, or HGTs, or in Europe, LGTs, lateral gene transfers, from prokaryotes. And these foreign genes make up nearly 1% of their gene inventory. And so previous work from our lab and collaborators, which is this top paper here, has shown bioinformatically that many of these HGTs encode adaptive functions related to polyextremophily. And these include metal and xenobiotic resistance and detoxification, cellular oxidant reduction, carbon metabolism, amino acid metabolism, osmotic resistance, salt tolerance, and salt tolerance. And so we have a lot of projects focusing on a lot of these things, because there's a lot going on here, but it's all part of this highly collaborative project between our lab, other labs, NASA, and the Joint Genome Institute, where we're producing these like really big lab and field-based multi-omics data sets to try to characterize the Cyanidiophyceae and how they interact with the other microbes in their community. And so part of this work was to generate these large metagenome, metatranscriptome, and metabolomics data sets from samples that were collected from the microhabitats I discussed at Lemonade Creek in Yellowstone National Park. And so today I'll be focusing on some of the metagenome data, which was, the first round was analyzed by a postdoc in our lab named Tim. And he was able to identify from all of this microbial data 2 isolates that matched reference genomes that we had already sequenced, which were Galderia sulfuraria 5587 and Cyanidiophyceaen maroli 10D. And we also found over 100 different prokaryotic bins, which include a bunch of archaea and various different bacterial lineages. And so, you know, there are some larger aims of this project, which are to study detoxification and transition between autotrophy and heterotrophy, because these algae, or the Galderia at least, are mixotrophic. But I'm going to be focusing today on this project that I'm working on right now, where I'm looking to identify putative prokaryotic donor lineages that live alongside the Cyanidiophyceae so that I can model how gene sharing occurs within this environment. And so right now I'm applying alignment-free K-mer-based methods to eukaryotic and prokaryotic sequences to identify regions of foreign gene transfer. Oops. And so horizontal gene transfer it's the end to a long process, basically. So the beginning of this process is the integration of a maybe large chunk of foreign DNA, which will then be altered by selection. And most of these chunks will be purged from the population very quickly. But if there's a gene in there that encodes a beneficial or adaptive function, it may persist while other parts of the original transfer go away. But fragments of that original transfer might remain in the genome nearby or elsewhere, and they might give us some clues about HGTs, like of transfers received in the past. And so since uptake of foreign genetic material is not biased towards genes as the unit of transfer, we predicted that foreign DNA integration is occurring at the DNA level and not the gene level. And we also hypothesized that by using a large metagenomic data set like our Yellowstone data set, we can make connections between foreign DNA regions and their donor genomes, and that these organisms live in close association with one another. And we predict that the flow of DNA involving the red algae is mostly from the prokaryote to the eukaryote. And so to look at all of this like transfer in action, I've begun working with a tool that was developed in prokaryotes, which I guess I'll just explain a little bit. So this is an alignment-free tool that employs the generation of K-mer profiles across genomes or scaffold sequences and a statistic called term frequency inverse document frequency, or TF-IDF. And this was originally, it's really cool. Like, it was originally developed for the field of documents analysis in order to find words or phrases that are unique or uncommon in one document, but more frequent in like the broader corpus of documents. Or in a document database. And so it's been adapted since then by some of our collaborators to work on bacterial DNA sequences. And so the program that I've been using to do this was written by our collaborators at University of Queensland in Australia. And although they are eukaryotes, the Cyanidiophyceae have compact gene-rich genomes and they have little non-coding DNA. So like my assumption here is that genome-wise, I can use these organisms in the same program as we'd use with prokaryotes, and I should get like valid results. And so our lab is interested in utilizing K-mer methods for a variety of projects. And just like a little background is that K-mers are substrings of length K contained within a larger sequence. So that could be words composed of letters within something like a book, or that could be substrings of nucleotides of a certain length within a larger nucleic acid sequence. And so here I've shown an example of the word rhodophyte broken into its component k-mers of different lengths. So k-mers of length 4 or 4 mers, and then the same word broken down into 3 mers. Oops. And so all of the k-mers that make up a word or a sequence represent that sequence's k-mer profile, and k-mer profiles can be compared between sequences. And then for the example on this slide, which is a little messy, there's a brown genome on the top that's very different from the blue genome below it. And we can assume the brown genome exists as one of many closely related genomes or sequences in a database. And the blue genome is also one of many related sequences in the same database. Once we identify genomes or sequences of interest, we can build their k-mer profiles and then compare the profiles between the groups of sequences. And so using this TF-IDF statistic, we identify words or k-mers that have a low frequency in the brown genome, but a higher frequency in the blue and blue-related genomes. And then we can extract those k-mers from the genomes and inspect further to see if we think a horizontal transfer has occurred. So for example, if you break down the brown sequence into substrings of length 4, you're going to see a lot of the string LOVE, or the word love. And then you're going to see one hate string, which is right in the middle. But the blue genome has a bunch of hate strings. So we would then infer...
Yeah, we have 2 minutes, 3 minutes,
okay.
2 minutes.
Okay, yep. We would then infer that there was a foreign acquisition from the blue to the brown genome of this phrase that I've highlighted in red. And so we asked, can we use the TF-IDF statistic to identify unknown regions of foreign transfer, and can these methods be applied to our Yellowstone data set to identify putative prokaryotic DNA donors? And then can we then model how DNA flows within a community from genome to genome? And so far, I've just tackled this first question, and I have some interesting results. So by running this program between the reference genomes we already have for the Gaultieria and Cyanidiophyceaen, I was able to detect over 100 instances of putative genetic transfer from prokaryotes to the algae from 6 different donor lineages, which include 5 different bacteria, which are shown up here, and then 1 archaeon. And this analysis won't pick up previously detected HGTs because they were detected using orthogroup analysis, which works on the amino acid level. And although amino acids may remain the same, over millions of years, the nucleotide code below it may change, and it's not really compatible with the k-mer methods that we're using. But interestingly, I just wanted to highlight that as we expected, all of our results except for 1 were located in intergenic regions, and most are part of a cluster of short, non-repetitive sequences that are dispersed across the 1 to 5, typically kilobase intergenic region. And all putative transfers within each region were from the same donor lineage. So this indicates that these regions represent old and large transfers that have become non-functional, although some parts remain similar enough to the prokaryotic donor sequences that we can pick them up through this program that we're using. We also found one that was within a gene, and it's a gene that's unannotated. So it's possible that there was a functional HGT that has some sort of function that allowed it to remain in the genome for millions of years. We just don't know the function. I also ran this program on the scaffolds, not just the reference genomes, and I got like a lot more results. So right now we're just trying to optimize our filtering methods because a lot of this is probably some false positives. But anyway, there's just like a lot of data to parse through for this project. But from what we have so far, because it's still a new project, basically all of our predictions were apparent in the results that we generated, but the project is still new. And so going forward, we're just gonna skip through this slide, but going forward, I would like to sort through the results of just the prokaryotes and understand all of the within domain transfers that have occurred. I'd also like to take a look at the few Cyanidiophyceae to prokaryote transfer results and see if those pass filtering and see if there is any interesting transfers there. And I also really want to extend this analysis to our virus data set. Just one of our postdocs is still assembling the viral mags. And so, yeah, the ultimate goal here would be to just build genetic transfer networks to show how DNA has flowed through this community over evolutionary time. So that's basically it. So With that, I just want to thank my lab and NASA for funding my PhD and the JGI for funding all of our sequencing. And I will take questions.
Okay, thank you, Julia, for interesting talk. We have the questions on the chat. I will read them. From Marta, what are the main factors responsible for such a high genome content in the genomes of extremophilic eukaryotes?
So I guess just generally speaking, a lot of extremophiles end up having reduced genomes. There's a lot of like harsh stuff going on in these environments that could be damaging to DNA, and they often are just you know, over time they're retaining these beneficial genes, and the other ones are being purged from the genome. They're keeping what they really need. They're optimizing their genomes for only what they need and not a lot of accessory functions like mesophilic organisms might have the luxury of keeping in other environments.
Okay. The next question is, Julia, can you, is from Priya. Julia, can you tell me about the archaeon you looked at?
Yeah, so not like, I don't have details on that. It was in the family Thermoplasmataceae, and it was a genus that is not characterized or named. So it was just, it had a reference hit in one of the databases that we used. I don't know specifically what archaeon it was. I haven't actually, this is like, these are very new results on a new project. So I haven't like taken the dive deeper into these results yet.
Okay, the next is also from Priya. Also, what databases are you used for searching? And the second part of the question, are you looking at complete or incomplete genomes or all sequences databases?
Right. So are you I don't know if, are you asking like the databases we're using to identify the mags, or like the database we're using to identify the transfers? Because the transfers comes from our own database that we've curated based on our results from the environmental sequencing data. And then to like putatively identify the mags that we found, we use like a combination of like NCBI and like, I think a lot of other databases. I actually didn't do the binning for this project, so I would have to double check on exactly which databases were used to do the binning. But we usually are pretty extensive with it.
And the question is this also, did you use the complete genomes only?
The complete genomes only. I'm not quite sure, like
for your searching.
Yeah, when you're doing a search, so if you're blasting a database and you're looking at genomes that are not complete, you may find partials. You may find that the genome has been sequenced multiple times. So you may come up with 3 things that might have actually just been from one organism. So.
Oh yeah, no. So yeah, so I think that we do a lot of filtering. So like in the beginning, yeah, we kind of do everything, but by the time it's getting to this level of results, it's usually full complete genomes with good matches to our mags. I think that's what you were asking.
Okay, we have also one from Marta, and we need to finish. Is it like the non-coding DNA just get lost when the DNA gets damaged because of the harsh environment?
I mean, I think this takes a much longer answer to answer this question than the time we have. But I think, you know, over time with selective pressures, genes can change and become like pseudogenes and become non-coding if they're not so important. And it just, I don't know how to explain this quickly, but there's the whole Muller's ratchet process where these genomes just become very streamlined over time, and they're they're on this path towards becoming smaller and smaller due to pressures that produce constraints due to the environment they're in. Like a very summarized version of that whole process.
Okay, we don't have time for more questions and for the discussion. Sorry for this. We need to finish that.